Welcome, welcome. This is part 2, cell unit. So this is what we're going to talk about: the cell membrane structure and function, and discuss the different types of transport that occur across the cell membrane. So all biological membranes, whether it's a plasma membrane that separates the cell's internal environment from external, or membranes that surround the organelles and create these compartments within eukaryotic cells, they all are going to be composed of a phospholipid bilayer and embedded proteins. In addition to that, animal cells will also have cholesterol that's embedded within that membrane. So scientists often describe membranes as fluid mosaic because membranes are fluid and very, very dynamic. If we take a look at these phospholipids, we're going to see that they interact with one another in different ways. So the heads are hydrophilic, so they will be interacting with each other via hydrogen bonds. And also they are attracted to water, which is outside the cell, and water here inside the cell. So it means the hydrophilic heads will have high affinity to water via hydrogen bonds, and hydrophobic tails are going to aggregate on the inside of this layer. So, and then phospholipids can actually move laterally, and every now and then they can even flip-flop. So now if we embed proteins within the membrane, we're going to see that these proteins can also migrate throughout the plane of the membrane. This one is here an experiment that was done by fusing a mouse cell and a human cell. So proteins from each cell were labeled, and then when those two cells were fused, what we saw as the end result here is that these proteins were migrating throughout the plane of the membrane independently. So the membrane fluidity is influenced by the actual lipid composition and temperature. So if the membrane is made up of unsaturated hydrocarbon tails, you can see they have that kink or bend in the leg in that chain because of double bonds between carbon that happen in the hydrocarbon chain. This type of membrane will be very fluid. So these unsaturated fats basically enhance fluidity. However, if the membrane consists of saturated hydrocarbon tail, notice they are able to pack together because all the bonds between carbon atoms in the hydrocarbon chains are going to have single bonds. So therefore, these tails are able to pack together, and therefore this type of membrane would be less fluid. So now cholesterol that's present within the animal cell membrane is going to modulate the fluidity of the membrane by reducing fluidity when the temperatures rise and prevent solidification when the temperatures drop. So for example, fish at cold temperatures have more unsaturated fats to prevent the packing and remain fluid. So they basically can change and alter that fatty acid composition in response to temperature. So now let's take a look at the structure of the phospholipid. So you guys already know that they are considered to be amphipathic because they have two regions. So we have a hydrophilic region and hydrophobic region. So hydrophilic region contains choline, phosphate, and glycerol groups, and then hydrophobic region, so these are the fatty acids. So keep in mind that membranes are never symmetrical. So if we look at the two leaflets, one leaflet is going to be different from the other. As you can see here, we have an example of a little snippet of the plasma membrane, and here's the extracellular environment, and we have cytosol here, so outside and inside of the cell. And phospholipids actually come in different varieties. So we have phosphatidylcholine, here's one example. We have phosphatidylserine, and so on and so on. So now if you notice, the composition of the top leaflet is different from the composition of the bottom leaflet. So this is why we say these layers are asymmetrical, and the reason so is because they face different environments. So they have different things to deal with. And the same thing is happening here if we look at the endoplasmic reticulum membrane. You're going to see that that composition is also different from the plasma membrane because obviously they have different jobs and functions to carry out. So cholesterol, as I mentioned before, is an important component of animal cell membranes. Plants actually have a similar molecule called sterols. Hydroxyl groups in a cholesterol, notice, are here. And then the rest of the molecule is hydrophobic. So we have 4 rings and a short fatty acid chain. So this is why cholesterol is going to embed itself in a membrane in the following manner, where hydroxyl groups are going to be hanging out with the rest of the hydrophilic heads. And then the hydrophobic region will hang out with the hydrophobic tails. As I mentioned before, all biological membranes will also contain proteins within the membrane. So they come in different shapes and forms, and because they carry out different functions. So typically we can group them into 2 categories. So we have peripheral membrane proteins and also integral. So this would be an example of integral protein because you can see it's spanning the entire membrane. And then peripheral one is not spanning the membrane, so it's sitting below the membrane. So now understand that when we draw these proteins like that, like globular structures, you remember that they don't really look like that, because a protein is basically folded polypeptide, and a polypeptide is made up of amino acids. So you have a chain that's highly folded. And each chain of such will have a N-terminus and C-terminus. And then remember how they fold: secondary structure, tertiary structure, and some of them will even have a quaternary structure. So you can see here we have a protein that's called single pass protein. And this region is alpha helix. So you can see how it's spiral and secondary structure. In this case, the spiral alpha helix is providing a channel for specific molecules to go through. So it means proteins will be embedded in the membrane, and they will serve as channels, as pores, as carriers to move the molecules across the membrane. So if you can imagine, if I pulled this string right here, if I picked it up, I could literally unravel the entire protein into this long linear primary chain. And in some other cases, the protein is going to be spanning the membrane several times. So it will be going in and out, and sometimes you'll have multiple subunits that will come together and form these huge pores in the membrane. Some proteins, notice, attach to the lipid part and so on. So there is variation because they do carry specific functions within the cell. So now this is a cartoon drawing of a membrane. So spend some time here, make sure that you understand the structures. So now some of the functions of membrane proteins are as follows. So number 1 is transport. As you can see, molecules are going to be moving from one side of the membrane to the other, and some of the transport is going to be passive, some of it's going to be active using ATP. These proteins that are embedded in the membrane can also act as enzymes. Remember, they do not get used up by the reaction. They only help facilitate the reaction. So yes, enzymes can be embedded in the membrane. Some of these proteins will carry out a function of a receptor. So I'll call them little antennas because they are going to pick up signaling molecules, ligands, and then initiate a cascade of events inside the cell. So we call that signal transduction. Another major function of the proteins. For example, here we have a glycoprotein because we have that sugar chain that's attached. So this type of interaction, you can see this is one cell, that's a membrane, and that would be another cell that's coming in. So the other protein of that other cell is recognizing the sugar chain. So this type of interaction would be for cell recognition and usually short-lived. So this could be an immune cell checking another cell to make sure the cell belongs to that particular body and it's not a foreign cell. Intercellular joining, notice how 2 proteins are actually connected. So we're talking about these tight junctions that we discussed previously. And here we also have support as far as structural support, because some of the cytoskeleton can actually attach to peripheral proteins that are sitting within the membrane. All right, and now we're going to talk about the fact that membranes are selectively permeable, meaning they can regulate what type of molecules cross the membrane. As you can see here, oxygen, CO2, nitrogen, small hydrophobic molecules can pass the phospholipid layer directly, and so we say it has high permeability to these gases. So reduced permeability is for water and glycerol, some other components, for example, glucose and sucrose. These are large molecules, so they would not be able to cross the membrane directly. They would have to use specific carrier molecules, such as carrier proteins. And then ions, because they do have a charge, they will also they will not be able to cross the membrane, that phospholipid bilayer directly. They will need to utilize specific protein channels as well. So one example of the way molecules can move across the membrane would be passive transport. Just like the term suggests, this type of movement does not require energy because molecules are moving down the concentration gradient. So in other words, we have high concentration and they're moving to where they have low concentration. So they're going down that gradient. So we call this simple diffusion. Certain types of molecules, such as gases or small hydrophobic molecules, lipid-soluble molecules, can diffuse directly through the phospholipid bilayer. Larger molecules, hydrophilic or ions, they would have to use a specific protein channels, and we're going to talk about that in a bit. So now, notice that diffusion is happening from high concentration. So here we have all these molecules that are highly concentrated on this side of the membrane, and they are moving down a gradient, so it means they're crossing that membrane on the other side. And then the purple molecules here are moving the other way because you have high concentration here and then low concentration there. So then eventually equilibrium is going to be reached. Now, do not think that these molecules are going to stop moving. They are continue to move, but there will be no net gain or no net loss. So now, anytime a molecule is too large to cross or has a charge and is not able to cross the phospholipid bilayer directly, they're going to be using proteins. So we call these proteins channel proteins and carrier proteins. A channel protein would be an opening that will allow for a specific molecule to pass through. So examples would be aquaporins. So they allow for water to pass through, and then some other channels would be ion channels. So it means specific ions are going to be moving across that membrane. So carrier proteins also carry out passive transport where the energy is not required. However, the way they function is by when a molecule binds to the protein, they undergo what's called conformational change. So the binding triggers that conformational change, and then it allows the molecule to move across that membrane. So you can see the molecule binds here to the protein, and then the protein changes its shape, and it delivers that molecule inside the cell. So now an example of a channel would be this. So we have ligand-gated ion channel. So this is where protein is going to open up that channel in response to stimulus. So for example, we have a stimulus molecule, a ligand that binds to the protein. You can see it's right here, binding site. And then the protein undergoes conformational change, and as a result, it opens up the pore and it allows for the molecules to move through. So this is a ion channel, so it means ions are going to be diffusing inside the cell. So this is diffusion because we have high concentration of molecules here and low concentration right here. So you see this is passive transport, but it's happening through the pore, is happening through that channel. So another example for the carrier proteins would be glucose transporters. Now we do have different types of glucose transporters depending on where and what cells they are situated. But this one, you can find that in the actual neurons. So in the brain, and this is where the glucose is going to bind to the protein that's sitting within the membrane, and then the protein changes its shape in such a way that it releases that glucose on the other side. So notice this is passive diffusion because we go from high concentration to low, so we're going down the concentration gradient, and therefore no energy is required. So now, unlike the passive transport, active transport will require energy because we're going to be moving molecules against the concentration gradient, and this is where energy from ATP is going to be utilized. So it means we're going to have to hydrolyze the ATP, utilize that energy that's been released to power certain proteins. So we call these proteins pumps. They're able to move substances, molecules, from one side of the membrane to the other. So, and we have two types of active transport. Primary active transport. This will involve direct hydrolysis of ATP for energy, and these are your protein pumps that are going to pump the molecules against the concentration gradient. Number two, secondary active transport. So this one is dependent on primary active transport because it uses that electrical gradient that's going to be generated because of primary active transport. But then that gradient is going to drive the other molecules into the cell or out of the cell. All right, so let me give you an example of a primary active transport. So as I mentioned before, they're going to use ATP directly. So they're going to hydrolyze the ATP, and these proteins are generally referred to as pumps because they're going to be moving molecules across the membrane, and oftentimes they transport ions such as hydrogen, calcium. As you can see here, we have sodium and potassium. So because they are moving ions across the membrane, we will refer to them as electrogenic pumps. So which means they can generate membrane potential, meaning they can generate voltage across the membrane. So what do we mean by that? So let's take a look at this cell right here, which is an axon of the neuron. So now we're looking inside of this axon, so we call that cell interior. And notice the charge on the inside of this cell, we have a negative charge. You see all these negative signs? So, and then the outside of the cell is going to be positively charged. So anytime a neuron is not conducting an impulse, it's at rest or it has a resting potential, we say that the charge on the inside of that cell at this point is going to be negative. But when the charge is reversed, it becomes positively charged because of channels that have opened up and allowed for the influx of sodium. So now the charge is reversed, it becomes positive, and we say we have generated action potential. So all of these turn into positive charges here. And they happen obviously in sections. So this is the action potential. It cannot remain like this in the cell. So it means we have to restore the resting potential of the cell, of that neuron. So the way we restore it by using sodium-potassium pump. Because sodium rushed in and now depolarized the membrane, sodium-potassium pump is going to have to move that sodium out and also deliver potassium in. So it moves those ions in particular ratio. So 3 sodium ions will be pumped out and 2 potassium ions will be pumped in. So what this means, this pump generates a deficit of positive ions inside the cell. So if we have a deficit of positive ions inside the cell, we have restored the membrane potential, that resting potential, and we again will have that negative voltage inside the cell. So this is an example of primary active transport where a protein utilizes ATP, hydrolyzes it to actively move ions across the membrane. Secondary active transport is driven by the energy stored in the electrochemical gradient. So we can call that electrochemical gradient as proton motive force. So we actually have to generate the proton motive force first in order for the secondary active transport to take place. So what happens here is this proton pump is utilizing ATP, hydrolyzing it, to actively move hydrogen across the membrane. And you can see we accumulate this high concentration gradient of hydrogens on the other side of the membrane. And then what they do, they actually diffuse through another protein. And as they're diffusing, this force is going to drive the import of sucrose into the cell here. So you can see this particular transporter is called co-transporter because as the hydrogen is moving, sucrose is moving as well. Except that the hydrogen is diffusing, so it's going down its concentration gradient. So, but then this force, this electrochemical gradient, is allowing for the movement of sucrose against this concentration gradient. So this is what we identify as secondary active transport. So here's another example. We have electrochemical gradient that is generated by primary active transport. So it means we have a pump that actually moves the ion, and in this case the ion that's being moved, it's sodium. Secondary active transport or co-transport happens when we accumulate the high concentration gradient of sodium. The sodium will help move glucose across the membrane as well. So this carrier, this glucose carrier, is actually a sodium dependent carrier. So it means if we want to move the glucose across the membrane, that sodium concentration gradient, that electrochemical gradient, is going to drive the movement. So if one of these solutes is missing, the movement is not going to happen. So you can see glucose molecules are sitting in that binding site, and then you have sodium, they are occupying the binding site. So when they all are there, the protein will undergo conformational change, and it will allow for the movement of the components in the cell. So you can see how sodium drives the movement of glucose into the cell. So now we have different types of transporters. Is the uniporter, so it means this particular protein will carry a molecule or an ion just one at a time, one direction. If the protein is carrying two different molecules, like you saw in a previous example, but they're going in the same directions, we will call that type of transporter a symporter. And antiporter is where you have two different molecules, but they're going into two different directions. So just to give you an idea how complex things are and how highly regulated all the traffic is, here's a plant cell. So this is the outside of the cell, and then this is the membrane, you can see right here, and then we have a vacuole, and of course you would have all the other organelles, but they're not shown here. So you can see we have a number of symporters. So a few examples would be, here it is, sucrose is being moved and is driven by that hydrogen gradient. So amino acids can also be driven like that, the same way, hydrogen gradient. Peptides, meaning short sequences of amino acids, are being driven that way. So all of these are symporters. So basically the hydrogen gradient is dragging them along into the cell. So antiporters, notice they're moving molecules in two different directions. So here, when it comes to vacuole, notice the sucrose is being moved in the vacuole, and then the hydrogen is leaving it. So you can see they're going in the opposite directions. So now if you remember, we also talked about channels. So these channels would indicate facilitated diffusion, when molecules go from high concentration to low. No energy is required because they are going down the concentration gradient. And then we also have pumps. You can see here we have a pump that's moving hydrogen out, and it's utilizing ATP. So you see how much we have here going on? So all the traffic is very, very highly regulated. So now, exocytosis and endocytosis are going to be unique to eukaryotes only. And these processes occur because of the use of vesicles. So of course this will require energy and even motor proteins to be able to pull these vesicles, and dynamic cytoskeleton that we can build and break down. So exocytosis happens when a vesicle fuses with the membrane and excretes or secretes the contents out. Now cells can also use exocytosis to actually embed proteins in the cell membrane. So when they make a protein, that protein will be inside the vesicle, and then when the vesicle is fusing, that protein will be embedded in the membrane. And then also they do so to remove cytoplasmic waste. So 3 different types of endocytosis. would be phagocytosis. This is what we refer to as cellular eating. That means the membrane can actually form these folds you can see around the food molecule, or if it's white blood cell, it could be engulfing bacteria or any kind of pathogen. They will bring in that vacuole inside, and then of course that would fuse with lysosome to break the macromolecules down into smaller units. Penocytosis, you can see the formation of the plasma membrane is different because it forms a little sac and now it fills up with fluid. This is why I refer to penocytosis as cellular drinking. And receptor-mediated endocytosis is initiated when particular ligands bind to the receptors. So you can see we have these receptors that are sticking out on the surface of the cell. And then when these ligands, such as this, bind to the receptor, it triggers the formation of a vesicle to bring it inside. And here is a nice table that compares the different membrane transport mechanisms. All right, and I will see you in part 3 about osmosis and osmoregulation.